Hello. This is Mr. Tolbert's class. This is biology in Mr. Tolbert's class. This is lesson number 2. This is lesson number 2. Now we're going to talk about the transformation that happened as we are learning about DNA. Griffin reasoned that some chemical factor could change harmless bacteria into disease-causing bacteria and was transferred from the heat-killed cells of the S strain into the live cells of the R strain. At the bottom of the screen here, you see a picture. Now, here's the very first pictures were the heat-killed ones. Then they put it in a injection deal and injected into the mouse, and the mouse died. So, what happened there? This was to find out, how did the mouse die if we killed all the germs? He called this process transformation, because the type of bacteria would be changed permanently into another type of bacteria. So it's a groundbreaking thought process. From one type of bacteria that was totally benign and would not cause you to be sick, and it would turn into the kind that can kill. Because the ability to cause disease was inherited by the offspring of the transformed bacteria, Griffin concluded that the transformation factor had to be something that was passed down. It had to be a gene. Now let's get into the molecular cause of transformation. A group of scientists at the Rockefeller Institute of New York, led by a Canadian biologist by name of Oswald Avery, wanted to determine which molecule in the heat-killed bacteria was the most important for this specific transformation. And as you recall, that Griffin figured out that there was something there, some gene that was there that was passed on. So Oswald Avery was trying to figure out which exactly was that. We're trying to narrow that field down that caused the pneumonia that then killed the mice. Avery and his team extracted a mixture of various molecules from the heat-killed bacteria and treated this mixture with different things. Now, this is the cool thing. He took these enzymes and destroyed the proteins so that it could not be proteins that caused the transformation. They took certain enzymes that killed lipids, and lipids are fats, so these enzymes got rid of all the fats, and then that also, it's still transformed. Then they also put in an enzyme that would kill carbohydrates, thus rendering all of the normal macromolecules off the table for causing this strange transformation from benign bacteria to pneumonia-causing bacteria, where the transformation still occurred. He repeated it using the enzyme that would break down the actual DNA. The DNA is the last molecule that they hadn't tested yet. It's the last macromolecule. When they destroyed the DNA in the mixture of the transformation did not happen. Therefore, they figured that the DNA itself was the cause of the transforming factor. We'll watch that video in class. What role did bacteria viruses play in identifying genetic material? Here's another experiment. The Hershey-Chase experiment with bacteriophages confirmed Avery's results, convincing many scientists that DNA was the genetic material found in the genes, not just the viruses and the bacteria of all living cells. Several different scientists repeated these experiments. Alfred Hershey and Martha Chase performed most important experiments relating to Avery's discovery. It's called the Hershey and Chase studies, and they studied the virus on non-living particles that can infect living cells. Here's where we figured out that there is a certain type of bacteria called the bacteriophage, and this is crazy cool. Bacteriophage is what we use to do all our dirty work for us. It's a kind of virus that infects other bacteria. It's called a bacteria eater. Typical type of bacteriophage is what you see in this picture. we have figured out ways to add stuff inside of these bacteriophages. We use those now to literally change the DNA of things. This is how we learned to genetically modify the world, how we genetically modified the majority of the food and the plants that we eat now. When the bacteriophages enter the bacterium, it attacks the surface of the bacteria, attaches to the surface, and injects its stuff inside. So it injects its DNA stuff inside of the cell. The viral starts to make more and more of the bacteria from the inside, then splits open. Once it splits open, the virus bursts out. And that is the end of lesson 3. So now you'll need to make sure that you go when you take the lesson 2 quiz. So you need to take the lesson 2 quiz now, and I hope you've learned a lot.